Okay, so it's 11 o'clock now, and so it's my honor and pleasure to introduce everybody to the Plant Phenome Journal webinar series. I have not counted up how many we've done of these, but this is our little over 3 years now, so we're probably in the 30s. Really appreciate everybody bearing with us and attending during the current COVID issue. Also, I'll mention that I changed some things around given the recent sling of Zoom bombings. And so you need to log in with a valid Zoom user account. You're also muted on entry. And if you have any problems or questions, just type it in chat and I'll keep that open and keep maintaining that. And so if you do have any issues, please let me know or even send me an email. So today, the Plant Phenome Journal webinar for today is presented by Dr. Maria Monteros. She's an associate professor at the Nobel Research Institute. She's going to be talking to us about high-throughput approaches for phenotyping alfalfa germplasm under abiotic stress in the field. And I know at least one of her former personnel working on this paper has joined us. You can find that paper on the Plant Phenome Journal right now. After her presentation, we will open up for questions. Unlike normal, we won't unmute, but just type your questions in the chat box and either Dr. Monteros or myself can read them aloud. So without further ado, Dr. Monteros.
Well, good morning and actually good afternoon to some of us who might be joining us from Eastern time. I would like to start by thanking, first of all, Seth for the invitation to share some of our work. And as he mentioned, highlight that the first author on the paper, Bryce Kazanov, is actually on the call as well. So with that, I'll get started with my first slide, actually my second slide. Just whenever I start to talk about a project, we think about the background and the context for which the work describes can address a challenge or a problem. So if you think about the increased demand for agricultural products is going to continue to increase, thinking about challenges due to the access and availability and sometimes the cost of irrigation for growing crops, the inevitable challenge due to existing and emerging pest and plant diseases where oftentimes disease resistance genes are overcome by pathogens, thinking about conflicting demands and competing land use interests between agricultural production and urban expansion. Then also, oftentimes these marginal lands and soils have issues with nutrient limitations and acidity and present challenges for optimal crop growth. And that ultimately necessitates the use of fertilizers, but it increases production cost to address these deficiencies, and it can also lead to environmental impacts due to the excess use of fertilizer. For example, for phosphorus, plants only use up about 20 or 30%, and the excess ultimately runs off into waterways that end up in the ocean, creating dead zones and some other challenges. So in my team, we're interested in taking the scientific discovery and then bridging the gap towards crop improvement in ways that allow us to develop solutions to these challenges. And the topic today is alfalfa, and a quick background for those of you who may not be as familiar with the crop. So it's grown globally. It's a perennial species, meaning that if you plant it 1 year, it can continue to grow for multiple years. It's an outcrossing species, and it can be harvested multiple times per year. And the cultivars are actually synthetic populations, meaning that they're a composite of multiple parents. So I'll touch a little bit on that a little bit down the slide set. And as a forage legume, sometimes people wonder what a forage is. And it's ultimately a word to describe that it's really food for livestock, whether it's cattle, sheep, goat, horses, rabbits, anything. And as a legume, alfalfa has the capacity for symbiotic nitrogen fixation with rhizobia in the soil. And so it provides nitrogen for itself as well as for subsequent crops in a crop rotation system, for example, with corn. In terms of the forage production systems in Oklahoma, where our institute is located, it's largely based on winter wheat production and Bermuda grass in the summer. And by integrating alfalfa and some other species, including tall fescue, you can see where you can start to fill in those gaps and provide a year-round grazing system as part of this Forage 365 initiative. That's how this project initially got started. So thinking about alfalfa sources of variation, and actually originated in the area between Europe and Asia, and then certain types of alfalfa germplasm were introduced into the US at different years to bring in actually specific traits. So for example, the Medicago falcata provides cold tolerance, and it was used to expand the reach of alfalfa production towards more northern parts of the US. Our alfalfa breeding pipeline includes different steps, as people are familiar with defining your breeding goals and objectives, identifying germplasm, and then the next step is to select and breed to develop new populations. And in our institute, we actually think of it as developing testing and management plans for how it will work in agricultural systems that involves animals, the interaction with the soil and microorganisms in the soil, and ultimately get to the point where you can release, license, and market new cultivars with enhanced traits. So I'll start out with the first section of defining the breeding objectives and collecting germplasm. In the context of this project, looking at global acid soils and how this particular project aimed to look at abiotic stress and growing crops in low input conditions, you can see that these acid soils are an issue globally. And then within those acid soils in the US, you often get what's called acid soil syndrome that leads to deficiencies in other traits, and it's more predominant in the southeastern US with pH less than 6 in some regions. And then the deficiencies associated with nutrient limitations, for example, phosphorus. The existing reserves are dwindling, and so these phosphorus deficient soils are abundant throughout the US and require input of fertilizers or improvement of plants to uptake and utilize the available phosphorus. So this is kind of the background on our project. And then thinking also of the erratic rainfall and water adaptability, our low input conditions also included drought stress responses. This is a picture actually from Africa. So if you think of which gene pools you can access to bring in genetic variation, in terms of alfalfa, you can think of the Medicago sativa complex as our primary gene pool, and I'll touch base on that in a minute. And then you have your secondary gene pool and your tertiary gene pool, but for the emphasis on this project, we're going to focus on the Medicago sativa complex. And we have diploid and tetraploid forms. They're interfertile, and you can see that the different types have different types of flowers, different colors. And in terms of the diploid, if we cross the purple flower with the yellow flower, you get this sort of variegated flower. And in the tetraploid, which is what cultivated cultivars are, or tetraploid, you see that the purple flowered is more predominant in warm weather. And as I mentioned, the falcata, which is yellow flower, is more cold tolerant. Thinking about the diploid versus the tetraploid obviously has twice as many chromosomes, and so the plants are bigger, but the diploid has been used as a source of interesting traits, like some of the work that was done before in my team for aluminum tolerance in alfalfa. So thinking of more broadly of the composition of different species in terms of cultivars, soybean, you aim towards pure lines for genetic purity, uniformity. In maize, you have a hybrid system where you take advantage of heterosis. And in alfalfa, you actually have, as I mentioned, the synthetic population where you actually have diversity, and each individual seedling essentially is genetically potentially dissimilar from the other individuals within its same population. So we talk about populations in alfalfa rather than pure lines. in looking at potential germplasm sources, the U.S. National Plant Germplasm Sources is a pretty robust resource for germplasm. And ultimately, you want to evaluate those plants in the field for whatever trait you're after. This is a dedicated drought study. And then ultimately to find that variation where you can see that some plants are adapted, that they can grow well, that you can use for your next step, which is ultimately selecting the best plants, getting them either dug up, brought back to the greenhouse, making crosses, and then evaluate their productivity, whether in sward plots, as individual families, to determine which ones have the best trait, or recombine them, and we'll talk a little bit more about that. As I mentioned, we think about a testing and management plan that involves how the animal is going to respond in terms of grazing. One interesting story about alfalfa is that if you put it in a mix with Bermuda grass, the cattle will actually graze through the rows of alfalfa because it has really great palatability and it's a high protein forage. So then ultimately, this is where we're after, feeding our consumer and making them, enable them to put on some weight and provide better products. So thinking about the overview of my team, we have our trait development and breeding pipeline overview. So we span basic, translational, and applied research that involves genomics, transcriptomics. And for this talk, I'm actually going to focus on the section on phenotyping. We do things in the lab. We've done root evaluations in the greenhouse and looking at things in the field using remote sensing technologies, and ultimately to find beneficial traits and understand the genetics of those traits that can be used for breeding, prediction, making decisions, and get to better cultivars. Okay, so thinking about our field site, we wanted to identify a site that had low pH, low phosphorus. So we looked around on our campus, and you can see this image. This is our main campus in Oklahoma. And then we have what's called Research Park across the street. This is a linear irrigation system, but just up north of that, there was a potential site that we looked at. And the objectives of the project were to compare the phenotypic data collected from the field using remote sensing technologies with unmanned or unoccupied aerial vehicle sensors and manual approaches, and then rank the productivity of these accessions under low input conditions. So this plot area, if you will, shown in blue, was sampled in 20 different sections. I'm just showing a subset here. And the row at the top shows the pH, which is certainly acidic. Optimal conditions for alfalfa are at least pH 6 to 7, and these ranged from 5 to 5.6. And then underneath in the P by pounds per acre, it shows the level of phosphorus content in the soil. And alfalfa needs 60 to 65 pounds per acre for optimal growth. So after an initial soil sampling, we did a soil core sampling with these long sort of plastic tubes that had a plastic sleeve underneath at 122 centimeters depth soil core. And we broke it up into sections for these 4 subplots, if you will, 8, 9, 18, and 19. And these are the values for the different depths of coverage in terms of each of the plots. So we wanted to ensure that the phosphorus pH limitation would extend beyond the original subsurface soil. Thinking of the screening of global germplasm of alfalfa germplasm, the trial included 100 accessions, and I'll describe a little bit more of those details in a moment. But we wanted to look at plants that had high yield, tolerance to aluminum, acid soil, that were efficient in phosphorus, and we also included some lines from South America that were criolla or grown locally, into ultimately having 4,000 plants. Part of the reason why we wanted to look at South America is if you look at the atlas of the biosphere in terms of soil pH, the red color in this heat map shows strongly acidic, and that's in parts of Brazil, Bolivia, Argentina, etc. So these are some of the lines that we screened from those countries. As I mentioned, Brazil, Bolivia, Argentina, and some others from the US were also included. This is how we started. The seedlings in the greenhouse, they were germinated and then established under these domes for cover. They were also inoculated with rhizobia, a commercial mix of different isolates, and then they were grown in the greenhouse prior to transplanting. Then they went into the field, and you can see based on this diagram on the bottom left, there were 4 reps. Each rep consisted of the 100 accessions that were the 95 PIs or plant introductions, and 5 cultivars, and each of those little dots that you might be able to see represents a single plant. So each row consisted of 10 plants per plot. With 4 reps, that would be 4,000 plants. This is when the field was established. We had our slides to lay it out. And then this is what the field looked like in the bottom pane here the following summer. So you can see we had pretty good establishment, lots of variation that you can see from the established plants. And we also did a border row around the plot area of a cultivar called Bulldog 805. So thinking about phenotyping strategies using remote sensing, we combined manual analysis, and I mentioned it was for low input conditions, so there was no fertilizer, no irrigation, no insecticide, no herbicide, no integrated pest management. The only thing that was done was manual weeding in order to provide quality of plant images to distinguish between the plant and this ground versus having weeds that could confound those types of analysis. And then combine that with the UAVs and sensors that were mounted on a tractor, and I'll talk about those in detail momentarily. Okay, so the data that was collected include the number of plants, plant height, ground coverage, visual rating for growth, and the percent of the plants flowering. We also did above ground biomass sampling 4 times per year in the summer, and also phosphorus content, but I'm not showing those results today. And this is a sampling of the video. Hopefully you can see on the bottom left of the drone actually flying over the field area. And then on the right, you can see the spider tractor, which is a high clearance tractor, actually driving over the field, just to give you an idea of how long it takes and how the system would work. So this is described in detail on our manuscript published in the Plant Phenome Journal and the references there. So moving on to the remote sensing approaches, the UAV components include different types of cameras. This is the type of drone that was used. It had a red, green, blue, or RGB camera, and then also NDVI camera for multispectral sensing. And the payload here was about 160 grams. And then as part of the navigation of the software, this is a screenshot from the tablet in the middle of your screen. For the MISHIP setup, it actually gives you a feedback view, the parameters that were used, whether that's altitude, speed, etc. And ultimately, the remote pilot needed to be able to do this based on FAA or Federal Aviation Administration regulations. And then you can see this aerial shot here. We had 4 ground control points, one in the middle of the field and one around each of the corners of the periphery. And each of those points had GPS coordinates that were very clearly defined. And this is a sample of what it looked like, sort of white, black, white, black. And that will come into play later. So the traits that were collected weekly include aerial RGB images and DVI images, and thermal images. The models included NDVI, ground coverage, and estimates of dry weight, and it also looking at survivors and plant height with some factors. So why NDVI? Actually, the NDVI is calculated using different wavelengths within the near infrared and visible region of the spectrum. So it ultimately relates to the chlorophyll content of the plants based on the absorption properties to reflect the photosynthetic capacity of the plant and ultimately reflects the plant health. So the greener the plant is, a higher NDVI value, and if a plant is more stressed, like the picture here, you can see some a little bit of stress in the coloration of the leaves, that would have lower NDVI values. So without having to scout for each of the traits, whether it's disease or nutrient deficiencies, you can get a sense of the status of the plant pretty quickly. Okay, so moving on to the image processing pipeline. After a UAV flight, you acquire the different images after the flight. And then this next step is to stitch those images together using the geocorrection with those 5 ground control points and the defined GBS coordinates. And then the next step is to define the area of interest. So you would remove, for instance, the truck and the cars with this fishnet system that tells the location and the position of each row plot and the individual plants within a plot represented by each of those individual dots here. Okay, so you define the area, you pull it out, and then you calculate the NDVI, and then you create this orthomosaic image that's unfiltered. So in this case, it still includes the background, the temperature of the soil. In this case, it's the vegetation index. And then after filtering, you remove the background, and then you essentially can create this metric of NDVI units to reflect the plant health of each individual plot within the area. So after that, you do the calculation, you combine the NDVI images with the fishnet to ultimately extract the image information into an Excel file that gives you quantitative numbers for each of the reps, the row, and the plot, and then you can pursue with data analysis. And all this process is done within the ArcGIS software as described in our manuscript. And this is the outcome of what you get, the orthomosaic image for the red, green, brown. And you can see each of the dots or each of the plots represented by each of the individual plants. Most of them still have the 10. We did have some losses over the life of the stand. And then the NDVI, pre-filtering and post-filtering with each of the ratings determining the anticipated NDVI status. Okay, so moving on to the Phenomobile, and that's essentially any kind of movable phenotyping device. This is the spider tractor that has a high clearance, and it's a gasoline-powered tractor with a GPS system. It also has here the green seeker. It's a sensor for providing NDVI values that emits burst of light, and then measures light that's reflected back from the plant, and then the multi-spectral sensors here. And these actually provide information on photosynthetic activity, vegetation index, canopy temperature, etc. And then some ultrasonic distance sensors and the time of flight sensors can provide metrics of plant height. And then this was used, as I mentioned, for NDVI, canopy temperature, plant height, and then estimates to determine biomass and crude protein. This is an image depicting the AgriFogger software, and it's a custom software application that was developed at Noble Research Institute that allows us to retrieve, fuse, and write the data in real time to a CSV file. And that way you can process and extract the data on a per plot basis, essentially import it into an Excel type of system. So this is the output from the sensors that were mounted on the spider. So in addition to the Excel plot, you can see here the roadmap. It's zoom in. You can see each of the dots represents the plot, and the spatial resolution is actually pretty good. You can see when the rows were a little bit crooked. And ultimately, these models for the height, the protein, and biomass were based on the models for traits that were developed by Jason Pittman in 2015 to determine these traits for Bermuda grass, alfalfa, and wheat using these sensor technologies. So the reference is here. Okay, now moving into some of the results, you can present data in a clustering of accessions based on the harvested biomass, and this is data from the first harvest. On the x-axis, you have your entry IDs from 1 to 100, and then on the y-axis, you have the dry biomass as kilograms per plot, and this k-means clustering ultimately partitions the data into k number of clusters based on the relative distance between the observations So it's similar to like a multiple observation comparison, like Tukey's type of statistical analysis. And this is implemented in R by Bryce, and it actually generates these clusters to show you the different groups of accessions. So we also looked at harvest 1, 2, 3, and 4, and you can see that the number of clusters varies and was analyzed separately for each of the harvests. And then this is the total biomass combined for the year. And you can see there's 5 clusters, and ultimately, from a breeding perspective, you're already thinking that the ones shown up here to the right in blue are probably the ones that you want to look at for your breeding program. Another way to represent data is to look at just cumulative yields from the harvest. And you can see that there's a little bit of variation with high and low across harvests, but overall the ones on the right side of the screen are the ones that have the highest biomass that you'd want to use for your population development. So then looking at the heat map correlations between manual data versus remote sensing technology, on the top here is the manual data based on the kilograms per plot, and then correlating that with the data obtained from the spider or the Phenomobile versus the UAV data. And there were actually traits that were combined based on traits obtained from the spider sensor data and the UAV data. You can see across the board, based on the blue color, that shows higher correlation with the different traits. So there's a lot of traits like the, you can see the NDVI values, the chlorophyll content values that actually correlate pretty good with the biomass, as well as the ground cover and estimated dry weight here from the UAV data, indicating that they could be pretty good metrics of our ability to predict the dry weight of the plot without having to harvest. So you could essentially just mow the plot. And then since this project did include multiple harvests, the correlations were done separately and then combined for each of the harvests. And you see a similar pattern here. This is showing harvest 1 and harvest 2. And then after publication, we were able to continue harvesting this data. And so this figure here includes 8 harvests, 4 in 2 years. And you can see here on the right, these are the individuals that we would be most interested in. And then the same phenomenon when you do correlations from year to year, that it would allow you to use those models from prediction. And you actually see that in the 2018 data, the correlation values are a little bit higher in terms of the kilograms per plot, as some of these models are tweaked in terms of their ability to predict the phenotype. So since then, this is what the field actually looked like last year. You can see a lot of variation in terms of productivity. And down here, I'm just showing an example of this 41 plot. That's a PI from the US. This is number 100, is that Bulldog 805 cultivar. That some of the plants are looking good, but you've lost quite a bit of plants after 3 years. And then the line 79, which is a criolla material from South America. And then line 36 is actually a different PI from the US. So in October of that year, we sampled tissue for DNA extraction, and we're using genotyping by sequencing to determine genome-wide association studies, and that work is ongoing. And ultimately what that does, it just shows you here on the x-axis, each of the colors represents a different chromosome, and each of the dots represents a sequence variant that can be used to predict the phenotype. And then you can dig up the top performers that were used for crossing, and this is a constellation plot. It's actually pretty neat that shows you the different traits that are considered together. And it's a really quick visual way to determine which group of plants are high yielding that you definitely want to bring in for your crossing block versus low yielding plants that aren't as strong. So the next step is those survivors and those best performers were dug up from the field, and you can see here we're relying on our little friends, our bees. in bee cages, or we can do manual crosses. This is a close-up. You can see here the pollen tripping the flower to collect the pollen, and you can do manual crossing. And ultimately, this is what the seed pod for alfalfa looks like to generate new populations. And then coming back to the breeding pipeline for multiple cycles of selection, where you develop a population, you evaluate the plants as an individual or a family basis, then you select the best plants, develop new populations as a cyclical process, and then ultimately move into, once you have some populations that look promising, field testing for multiple years. And this is where our remote sensing approaches can really be useful in cutting down the time and effort that's needed to collect multiple data points in real time. This is all non-destructively and help us achieve the goal of developing an improved cultivar. Further, this is part of a broader scheme that includes the Alfalfa Breeders Toolbox, and you can search here by germplasm if you want information on a particular PI or your favorite genotype. You can search it, and if we have any kind of field data or phenotypic data, you can find it, and it allows you to search by gene, by sequence, different types of markers. And this is just the overview of how the phenotypic data for novel germplasm and some mapping populations, as I described, that phenotype with remote sensing approaches, when combined with a genome sequence, marker data, and SNP genotyping, gene expression data, can ultimately allow us to find new germplasm, evaluate plant performance, select plants for breeding, identify key genes, develop markers for selection, track those alleles through the breeding process, and then develop new cultivars as well as new knowledge. And then ultimately coming back to our pipeline where you are using technology-enabled approaches, whether it's remote sensing or a genetics and genomics toolbox to help facilitate the breeding process. And then just to summarize how this technology-driven alfalfa breeding pipeline, and I'm showing here the combination of the different technologies within plant breeding applications with genomics to identify genetic variation for the key traits based on your breeding objectives, and ultimately conclude that remote sensing is highly correlated with manually harvested biomass. These technologies can be used to detect genetic variation for traits. So we were successful for field phenotyping, for abiotic stress responses, and can be used as a tool for selection and breeding in alfalfa, and could readily be applied to other plant species, ultimately to develop better products that we can put in growers' hands. So with that, I'll just like to acknowledge, of course, Bryce, who's the main author of the paper, and our co-authors shown here with the asterisk, Push, Teresa, Mike, and Justin, who are part of the geospatial systems team and include some of our pilots, and then the members of my team. Bryce is shown right here, that helped with the planting, weeding, harvesting, field data collection, and our collaborations with the breeding, agronomy, and field plots team, as well as acknowledge, of course, Seth for the invitation and the TPPJ Journal for the opportunity to share our work, and our funding agencies, USDA and Noble Research Institute. So with that, I'll just say thank you, and hopefully we have enough time for questions. And again, here is the reference for our paper. And I would also like to direct you to our website, www.noble.org, where we have any opportunities available for postdocs, for students, for a summer scholar program, any kind of computing, data analytics. If you're interested, check it out, and you can also email me. I'm available online, or my email is available online. If you have any questions, please reach out. So that's all I have. Seth, if you want to take over.
Okay, well, thank you. And clapping on Zoom is still really awkward and weird. We've never figured out a good way to do that. That was great. I really appreciate how you covered how your tools are being applied to breeding. I don't think there's very many people I've seen who have sort of presented on that as opposed to just the technology. So this was really good integration of tools into a breeding program. So what we're going to do is open it up for questions. You got plenty of time. And what I'm going to suggest is that you, again, because of the Zoom bombing issues that some other societies have had recently, go ahead and write your comments into the chat section. If you prefer that either Dr. Monteros or myself read them aloud, you can direct them to us, or otherwise, if they can be public, just direct them to everybody and we will read them. So again, thanks for plugging the Plant Phenome Journal. We really like to highlight this type of cutting edge phenomics, which I think you presented, and its application. I would be remiss if I didn't point out that Dan Northrup, the editor, or the technical editor who handled your article, actually happened to join us today. And if I had known that, I would have thrown him under the bus to help host you. But in the interim, let me ask you a question to get us started. So as I mentioned, I think the application of plant breeding was really great. What challenges have you found in trying to run your clearly applied breeding program while integrating this new technology?
Well, so one of the things that I think we're fortunate at Noble is that the team that I described here in my sort of next to last slide, from our sensor technologies and the geospatial systems, is that they can provide that day-to-day screening of our flights, running the tractors, and we're extremely grateful for their collaboration. I remember my mentor, Roger Borma, used to say that plant breeding is multidisciplinary. And at the time, he was referring to, physiology and plant pathology. And I think now that concept has expanded to include multidisciplinary, working with agronomists, engineers, database managers, software developers. And so we've been fortunate to be able to engage with those teams in a support group that we can have a conversation of what our goals and the goals of the project are, and then execute them collectively. And as I mentioned earlier, Bryce is integral in this part of the process, and he was communicating back and forth with the geospatial systems team to develop different models and predictions jointly as things moved forward with the project. Or one of the examples is that the first flights, there were a lot of shadows in the images, so troubleshooting together was adjusting the time of day that the flights would occur to try to minimize those shadows. So it's truly an integrated collaborative approach rather than just here are samples, you do it, and then send us the data back. And I think that's very helpful of a successful collaboration, that you're truly doing this as a team, troubleshooting and developing new things together, where if there's different needs, you can work together, or if there's opportunities, you can say, well, I can use that for a different application. So doing things very collaboratively.
Yeah, that's great. It's nice to have a large team to work with and to help support and provide mutual support back and forth. Now, Anju Biswas, I hope I pronounced that correctly, had a question. Can you please explain the NDVI a little bit, how we can correlate NDVI with biomass?
Right. So essentially, the NDVI is a reflection of the photosynthetic capacity of the plant. And I'm actually going back to that slide, and it actually just represents ultimately the status of the plant. You would think that the more actively photosynthesizing material that the plant has would actually enable it to be more productive. So ultimately, it's based on the spectral reflectance that these sensors get light back based on the plant status. And here's an example that it's based on this equation that considers the light from the near infrared versus what you get from the red spectrum of light versus or divided by the addition of that, and it creates an equation. And if you look at our paper that was published in the TPJ, it describes there's different equations by Thorpe and some others that you can calculate and see which ones would be more suitable for prediction of your phenotype that could relate to biomass yield. So it's almost like you would take a DNA marker where you have a variant in a DNA sequence to see if that difference in the nucleotide would predict the phenotype. This is similar to that, where you're determining the status of the plant, the overall health. Obviously, the more vigorous and more productive it's going to be in its photosynthetic machinery could indicate that it would be producing higher biomass. But these are all sort of surrogates for selection that would be predictive. So if you're, depending on the species that you work with, I would encourage you to look at developing prediction models and then testing them against manually harvested or collected data as a proof of concept before you just jump into collecting sensor data and doing predictions and selections based on that. Did that help?
I don't see any other questions currently in my chat box. Oh, one just popped up. Did you use RGB images to calculate any vegetation index to estimate biomass or crop status?
So I'm going to scroll through the slide deck and let the postdoc on this project, Bryce, comment a little bit on that. Bryce, can you chip in here?
I need to figure out where he is to unmute him. There we go. Okay. Oh, there we go.
Hello, everyone. Are you hearing me?
Yes.
Yes. So I have indeed different RGB traits which I have extracted. I use the RGB to extract the ground coverage, what I called at that time ground coverage, which is finally the canopy cover. And the idea for that is at the time I was extracting my data and developing all those models with the UAV team, we were not capable of extracting height data. And the reason why I wanted height data was because the tractor, what we call the spider at Noble, was using height and DVI to re-estimate the biomass. So I wanted to try to compare the spider and the drone by collecting the same type of data and re-estimate the same type of data. Because we were having some challenges of extracting properly and accurately height using the drone, using the RGB images, I was trying to find another type of trait that I was at that time able to extract. So finally, exactly what you see on the images on the slide, which is shown by Maria currently, I was finally extracting the ground coverage by removing all the background, all the soil, weeds, if we were still having weeds, which were not removed eventually manually, and calculating the canopy cover, so the area covered by the plant on the ground. And that was important for the following reason. We have two different types of alfalfa which can grow into the field. You have some alfalfa which are growing upright and they can reach 70 centimeters height, even 1 meter height. But you have also some alfalfa which are staying lodged onto the ground. So they might be very long and with a high biomass, but they are still staying on the ground. So even if we were to extract height, it might not finally give you much more information if they are long, but they are staying on the ground. So the canopy cover was kind of giving me another type of trait, and I managed successfully to create a new equation using this canopy cover extracted from the RGB images and the NDVI that Maria just mentioned previously to re-estimate the dry biomass. And then we have some other LAI type of equations which were published in the past by other authors like Saito or Thorpe. And I tried to use those equations and apply them to our study. And finally, I was interested in trying to get the plant volume. So for that, I use the height from the tractor and the ground coverage I had just explained from the RGB images from the drone to calculate the volume of plants, and again, try to see if they were correlated or not to the biomass of plant harvested during the study.
Great. Next question, how frequently, from Holly Lane, how frequently do you think data needs to be collected throughout the season to be optimal? It often seems tricky to balance temporal resolution with the labor of collecting more time points.
So for this particular field trial, because it was our first one really deploying sensor-based technologies as a proof of concept, during the growing season, the time points were collected weekly. Obviously, there was a little bit of variation, plus minus a few days based on whether it was raining or if it was too windy for the drone to fly. But it seemed that weekly was a lot. Ultimately, if you compare it to the biomass harvest, we would harvest 4 times during the growing season. So one time in May, June, July, and August. We did find it helpful to figure out for alfalfa as a perennial, having more data points allowed us to determine which were sort of the tricky points in the season for alfalfa production. So by having images and being able to go out to the field, we were able to determine when the plants were starting to have some effect due to cold damage, when they would have the spring regrowth. And if you think about diseases or emerging pests, the more data points that you have would allow you to determine what that integrated pest management, if you want to start applying any kind of preventive or chemicals to alleviate. For this trial, because we did not do any management on purpose as part of a low input, it wasn't helpful in terms of making decisions for management. But I know that some farmers and some growers that interact with Noble Research Institute, they're using these sensor-based technologies to monitor the fields if they start to see a potential outbreak for different fungal pathogens, if they start to see any kind of potential for nutrient deficiency. You can use these approaches to think about targeted application of fertilizers and so many other applications. So I would say it depends on what your goal is. If it's to make selections for an annual crop, I would do it more frequently. If it's a perennial crop, you can probably do it every 2 weeks or even once a month once you have your models developed. But great question.
So if I may complement Maria on what you have just said, regard of the physical harvest, obviously we could do that only once a month, and I fear that if I try to do that more often, my team would have entered into a strike. So it was only once a month. However, for the remote sensing, most of the time we are doing it weekly, but sometimes we are going to one every 2 weeks or once a month, and it was really determined by how fast the plants were growing. So all the image which were collected, all the data from the tractor were analyzed very frequently. It was not accumulating all the data, and before starting the paper, we started to analyze. No, it was really weekly analyzed by us, and we could see when it was too often and unnecessary, or if we could space a little bit the different flight or data collection. Also, we found out that by collecting the data throughout the whole year, even when we were out of the growing season, it allowed us to see in 2018 the impact of global warming and climate change, when alfalfa was finally ready for the first harvest 1 month earlier than what is usually done. So it was easier for us to be able to prepare the team because the plants started to look like ready. And also when we were between harvest 3 and harvest 4 each year, it's when we were starting to have many more deaths because the plants needed to regrow while they were submitted to a lot of stresses. And it's usually when the numbers of survivors was decreasing a little bit faster. And so flying the drone pretty frequently was allowing us to really stay in touch, if I may say, with the field.
Okay. We got a couple more questions here. Adrian Gracia Romero asks, have you estimated canopy temperature using a thermal sensor with the UAV? And have you compared it with canopy temperature estimated with your tractor?
I think I might answer this one, Maria. We were trying to get information from the thermal camera, but at that time, the software such as Pix4D and AGSoft were losing the temperature information when you were making an orthomosaic of multiple images collected throughout a flight. So if you were to take every single images at that time, you were in each image still having the temperature data. But then when you were creating an orthomosaic to have the full field, then you were losing them. The bypass would have been to fly higher, but then you would lose a lot of resolution, and we decided to not do that for what we were doing. So finally, at that time, we stopped. With my current knowledge and the technologies that I'm using in my current position, we are collecting thermal data very frequently, but at that time we did not.
Okay. I skipped over a question, another question from Anju Biswas. They actually have two questions. Why are examining family-wise instead of individual plant for everything? And then is there a facility for grad students to do short summer training for data analysis, such as GWAS or RGB data?
Okay, so if you think about alfalfa as an outcrossing species, we were able to look at individual data, but because of the breeding system that you are looking at populations rather than individual plants, and you use, let's say, 25, 50 parents for alfalfa, we wanted to identify the best families, and then from within the best families, select the best plants. So one of the things that you think about as a breeder is, are you going to take the best plant from a family that performs well, or are you better off taking, or what trade-offs are you considering when you look at an individual plant that looks really well, is productive, but all of the other plants within that family are struggling and not doing really well? So in short, you could look at individual plant bases. The technologies do provide you with the resolution. It was just a biological component of the alfalfa as a system that we looked at family versus individual plants. But we actually selected individual plants from within families to dig up and then make crosses in a greenhouse to develop new populations. But great point.
And your any training facilities?
Oh, yes. I'm sorry. So Noble Research Institute has what's called a Summer Scholar Program. And what that is the postdoctoral fellows at Noble will write proposals and then match them up with students that apply for that program. And it's a paid internship that Noble has often provided a stipend, housing, transportation to and from the airport, and you get a mentor experience to do a project. You get hands-on, whether it's in the lab, in the field, depends on your interest and the projects that are applied. Students get paired up with a mentor, and then you could present your project at the end of the summer before going back to school. So this would be for undergrads. We've also had interns or training programs. One of my team members, she's originally from Argentina. She came initially a number of years ago as a student just to learn about molecular resources. So if you're interested, I encourage you to email me or look at our website, www.noble.org. It's here underneath the thank you on this slide for more information. Hopefully things will return back to normal after the coronavirus situation that has us all currently working from home except essential functions. So thank you for your interest.
So great. Shay Simpson just says, thanks for a very nice presentation. It's interesting to see the differences in alfalfa breeding compared to other crops. Maria, can you please take a look at your chat and just make sure nobody directed anything to you? And then I'll ask a follow-up question to one of the things you sort of already answered, which is on your correlation matrix, the ground cover correlated extremely well with plant biomass. I know from one of the other articles that I edited, and I can't remember the crop off the top of my head, at some point when the plants, if they're not spaced plants, if they're actually planted fairly densely, at some point the plants start growing up rather than out. And so I wonder if you would expect to see that canopy cover still being so highly correlated if you hadn't done multiple clippings, for instance, or if you had planted it more densely.
Well, and this is something to think about as the plants move forward in the breeding pipeline. We move away from space plants into more sward type of systems, and that's where the plants get planted at a higher seeding rate, meaning that there's more plants within a given area to more closely represent what a rancher would put in their property. And so I would anticipate, as I've seen some studies in corn, where as the plants are spaced closer together or further apart, it affects how the plant grows in terms of the canopy, the root system, and how some of these traits will adjust and adapt to what ultimately you get what you select for. And so you want to make your selections from a practical perspective in the system as close to the actual growing conditions that the plants are going to grow. Because yeah, if you have plants that have more room to grow for you to evaluate its full genetic potential, the canopy cover of that plant versus an individual plant in a different system would be different. Could that plant adapt or have that plasticity that would allow it to grow and do well in a sward setting versus a space plant setting? So I would determine that empirically for each species and the spacing and field conditions for the plant.
And if I can complement a little bit, when I developed the model which extracted the canopy cover, or as I called it, ground coverage from the images, I was thinking also at that time that I would very quickly face some limitation of doing that because of the fact that the plants were only 1 foot apart within one row, 3 feet apart between rows, and that I would quickly have them going over each other. And finally, I did not really see that problem. And when checking my correlation at before the first harvest, when the plants were the tallest, versus later on during the subsequent harvest, when the plants were still shorter, even regrowing, it was not, the correlation was not decreasing that much. So it was pretty nice. However, what we have to keep in mind is even if the plants are growing and are becoming very long, and I'm saying long and not tall, they are not ultimately becoming taller. So we went to the field to measure manually the height of plants, and you were having the height which would be eventually, let's say, arriving to your knee for plants which were more than 1 meter, 1 meter 20 long, but they were only on the ground. So even if they were getting very long, they were not ultimately growing in height for some of them. So it could be a limited factor to estimate, but when I estimated my dry weight and I used ground coverage as one of the factors, it was still very highly correlated, taken separately per harvest or all the harvests together. And for the following years in 2018, I used the same equation and verified my model, let's say, and it was still very well correlated.
Great. Well, we've reached the noon hour. There's a lot of thank yous that everybody and compliments on the presentation. I'll just echo that. Great presentation. I like the tag team too. I'm glad you guys were able to be successful in this, and thank you again for publishing in the Plant Phenome Journal. I will put this recording up in the near future as long as nobody protests to that. And if there's any other further questions, I think Dr. Monteros put her email address in the chat box, and she's probably pretty easy to find. I found her pretty quick with a search to Noble Foundation. So thank you again, and I'm sure everybody is clapping simultaneously. Any last words, Dr. Monteros?
I think I'm good. Just thanks everybody for participating today. Thank you for organizing, and Bryce for his work and answering your questions.
Okay, and with that, everybody have a great day, and we'll look forward to seeing you on the